question that we need to be asking ourselves is, well, how do I know that we're coming out of an ice age and that we're coming out of a glacial period, and that these other periods of times, like the 1970s, which was cooler and wetter, only are going to last maybe a decade or 20 years, or that the time frames that were the Little Ice Age or were the younger, driest periods, only a few hundred years in duration. And the model that I just reproduced for you over a number of slides comes out of evidence of past climates that we can measure. And there are a number of different ways that we can look at that historic climate, test it against many different sources to determine its accuracy and its consistency. And we call that proxy data, a proximal data. We can't go and teleport in time with our thermometer and say, all right, we're 10,000 years in the past. I'm standing in Antarctica, or I'm standing in where Youngstown, Ohio is going to be, and I've got my temperature and in my thermometer, and I'm going to record it. That's just not possible. Jules Verne didn't give us instructions on how to build that time machine in his novels. So we have to figure out other ways to time travel through proxy data. And there are a number of different proxy data that get used to help us appreciate what those historic time frames looked like climatologically. The big ones, and there are several different proxy data sets, but the big ones are ice cores, lake and seabed cores, tree rings, pollen analysis, and coral. So with proxy data and ice cores, and we've got some great ice core samples here in the United States, starting with the Byrd Polar Research Center here in Ohio, is we've got ice cores from all over the world. And ice cores, like rock cores, can be dated. We can see layers in them. So what ends up happening is snow falls, and then if it does not melt over the summer season, then the next year the fresh snow falls on top of the old snow, and it ends up trapping in between the snow the atmospheric content of the time period that snow had fallen. Now, the snow, as it gets buried deeper and deeper over subsequent years of snowfall, will morph into ice, and that the ice bubbles will capture that historic atmospheric condition. So we're going to see things like oxygen ratios, CO2, methane, and other chemicals in the atmosphere, but we're also going to see particulate matter, to include pollen, to include sea salt. So you've got things like that. We can get evidence of volcanic eruptions in those layers of ice, and so we can date each one of those layers, take a look inside that layer and figure out what the chemical analysis is. And based on the ratio of oxygen 16 or oxygen 18 or carbon dioxide, we can then determine from actual observation, measure against what is being measured in the ice core with what has been observed elsewhere, what the temperatures were like. And so this data for ice cores have been collected everywhere from the Andes ice cores in the Himalayas to Greenland, Iceland, Patagonia. So we're getting both northern and southern hemisphere ice. We're also getting ice from the eastern and western hemisphere. So this is a big data collective, geographically distributed source set. The problem with ice is that it melts. So once you collect it, you've got to move it, right? You do not want to lose your data. But data can be lost anyway, and data can be lost long before somebody takes that ice core and measured it. So on warm periods where snow is melted over the summer, that year or years or decades or longer does not get captured. All right, so that's important. One really important measurement that I mentioned a minute ago was oxygen 16 and oxygen 18, and I see that there's a typo here. Please take a look at permit us to read. It says oxygen 16 and oxygen 8. There should be a 1 in front of that. I will make sure that that is changed in the slide deck. So the 16 and the 18 are relative to isotopes, and I want you to just make this math super, super easy, all right? We'll not get too bent out of shape about it. 16 is 2 larger than 18, all right? So 2 of anything can be measured as heavier, right? So consider 18 heavier than 16. If something is heavier, it's going to take a larger low pressure cell to lift it, which means that it's going to lift more 18 up when the temperatures are higher, when the low pressure cells are bigger. So under a warmer climate, we should have more ratio of oxygen 18 versus more oxygen 16. All right. So that is one of the primary chemicals that folks like Andrew Klein at Texas A&M and others globally are looking at, is what are the ratios of 16 to 18 in that ice. Other past climate evidence includes sea and lake bed cores similar to ice. What we're doing is we're getting material deposited year after year after year, but in this case it's material which is deposited at the bottom of nice big lake beds, and whatever the ecosystem is in that lake, the organic matter from those organisms after it dies are going to settle to the bottom of the lake. So if you have a nice warm climate, the lake is going to be warm. That means the ecosystem members in that lake are going to be adapted to a warmer lake temperature. So the years and decades and centuries when the climate is warmer, the organic matter is going to reflect in the stratigraphic layers in that lake bed warmer organisms. As time goes on and the temperature starts to cool, the ecosystem in that lake is going to transform to members of a cooler and cooler and cooler organism or series of organisms. So the material which then rains down from those organisms are going to be reflective in the stratigraphic layer of a cooler period as it starts to evolve and change. So just like the ice cores, we're going to dig into this. Lake beds usually go undisrupted, so we don't usually see gaps in the data sets that last significant amounts of time. So we generally have more complete data sets that allow us to see the regular transition of warm and cool cycles based on the organisms, the pollen, maybe some of the fossil evidence within these lake bed sediment cores. Tree cores, on the other hand, give us a more refined date that we can see. Now with tree cores, most of us recognize that tree rings are an annual condition where the center of the tree ring is the pith, and for about the first 8 years of that tree we have a pith. So we just sort of start out with saying, all right, this is 8 years, we can't really see the tree rings in here, so we count back. So if this was the ring that's forming this year in 2021, this ring here would be 2020, 2019, 18, 17, 16, and we count backwards into the center of the tree. Each tree ring based on its structure, will tell us what the season was like. Was it warm? Was it cool? Was it wet? Was it dry? And we have two functional rings that account for one ring. So oftentimes people will count the light and the dark, and they'll go 1, 2, 3, 4, 5, 6, and they've counted 6 rings, but there's only 3 years there. The light colored rings are the early spring summer growth. So out of the winter, we've got lots and lots of nutrients in the soil from the winter, from the fall, when leaves and other matter fell to the surface of the earth and started to decay. And as the winter snow melts, it takes all that decayed material, pulls it into the soil, and then the tree takes up that nutrients with the water and puts it into the cellular growth of the spring material. So the light colored ring is usually nice and thick because it's that great volume of nutrients that's coming from the winter and the fall. We get a period of time in the summer where there's retraction of growth. We go into a hiatus season in the summer, and in the fall, late summer and the fall, we get a renewed growth. Moisture returns back in the fall, and that plant starts to generate a new growth ring in the fall, which is thinner and darker generally. So what this affords the individuals who are looking at the climate data in trees is rather than centuries over time that you get in ice cores and lake bed cores, we get spring and summer evidence, or pardon me, spring and fall evidence. Late summer is true, but spring and fall evidence. So the data is much more refined, and we know that what particular tree species range of tolerance is. So we'll know that certain trees like it dry and cold. So we cut into that tree, or we produce a tree core, and we measure that tree core and say, all right, do the rings look like they should for a tree that likes it dry and cold? And if the core doesn't look really healthy, then we can analyze that core a little bit more clearly and see that it was maybe wetter, maybe warmer. And then we take that and we measure it against a tree that likes it warm and wet. And we see if those rings at the same time frame are actually really healthy and shows that the tree was doing well over those time frames.